Okay, welcome guys. So last week we had a look at the transition from DNA to mRNA, which is called transcription. And this week will be a little bit easier, and we're looking at the transition from mRNA to protein. So we can see this is the sort of timeline of events that we go from DNA to mRNA to a protein at the end. So what do we know so far? We know that transcription is the process of making messenger RNA, mRNA, from a template strand of DNA. And RNA polymerase is the super important enzyme that separates the coding in the template strand and then builds the RNA sequence along the template strand. And we can see that the mRNA nucleotides consist of A, U, G, and C, which has this new addition of a U, which replaces the T. And all of this occurs in the nucleus. So what we'll learn today is what a protein is, how mRNA is used to make a protein, and what the process is called, and how to decode a protein from an mRNA strand. So if we go from DNA to RNA, or mRNA in our case, this is called transcription. Now the new thing we're looking at today is RNA to protein, and this is called translation. So it's very easy to mix these two up, but the way I try to remember it is that the protein is translated from the RNA code, whereas the RNA is like written or it's transcribed from the DNA. So a little bit about proteins first. So like DNA, proteins are a polymer that are made up of monomers, and these monomers are called amino acids. So we can see our protein chain here, which is often called a polypeptide chain, but we'll just call it a protein chain for now. And it's made up of all these little balls, which look like beads on a piece of string. Now, each individual bead is an amino acid, and we can see an up close here of what they look like. We can see they've got little names on them as well, which we'll explain shortly. So there's 20 naturally occurring amino acids, and these all combine to make different proteins. So the order of these different amino acids will result in a different protein. Some examples of proteins are insulin, which is really important for your blood sugar levels, hemoglobin, which is what's vital for getting oxygen around your body, and enzymes. So RNA polymerase is an enzyme, so it's an example of a protein. So this is all of the different proteins, and we don't need to remember them. I don't know them, but we just need to sort of understand how we sort of symbolize them. So we can see that in this case, we've got one here called alanine, and it's got a single letter symbol and a three-letter symbol. Now, every single other one has this same thing, a single letter and a three-letter symbol. And we can see in the case of this, we often represent the amino acids by their three-letter symbol. So if we're ever trying to write down a specific amino acid, we write it down by the three-letter symbol. So let's get into translation. So this is translation, which is the process of getting from RNA to a mRNA to a protein. So this first step is that the mRNA needs to leave the nucleus, and the transcriptions happen in the nucleus, so it needs to get out of there. And what it needs to do is bind to one of these little blue dots. So it needs to get from here out into the cytoplasm and bind with one of these dots, which is called a ribosome. So what this is a ribosome up close, and it's a little factory, and what it does is it reads this piece of mRNA. So what happens is it gets sandwiched between these two subunits of the ribosome, and it sort of moves along like a conveyor belt, and this ribosome reads the code on the mRNA. And there's a specific way that it reads the code, which brings us to our next step. So step 2 is that the ribosome starts to read the mRNA strand. So the mRNA strand is read in groups of 3 called codons. So rather than just reading one nucleotide, it reads them 3 at a time. So right here, this group of 3 is called a codon. Same as this, each group of 3 is called just a codon. So it's a triplet code of nucleotide bases, and each of them will code for a specific amino acid. Each codon will code for a specific amino acid. So it'll make one of those beads on the chain added to make a protein. So after reading each codon, it will meet one of the following: UAA, UAG, or UGA. So these are each a codon. They're 3 different codons. And what they do is they tell the ribosome to stop making the protein And what will happen is it'll release the string of amino acids, and now we have a polypeptide chain, which is our protein chain, which is formed. So it will read each of these, and then it'll reach this last one, which is UAA, which we can see here. It tells it to stop, and then it'll release this protein. And also another one to mention is the start codon. This is the same for every single protein. So every single mRNA strand that's coding for a protein will start with AUG. And that's how our ribosome will recognize, okay, well, let's start making a protein. So how do we know which amino acid will be added? So we mentioned that a codon is the triplet code. So we can see that that's what the codon is. And each codon codes for a specific amino acid. So in this big chart here, we can see that we have the codon sequence here, and they're matching amino acid. And there's 64 different potential codons, because that's just the way that you can arrange all the amino acids, all the nucleotides to form 64 different ones. But we know that there's only 20 amino acids. So we'll say AAs. We can shorten it to AAs. So what will happen is you'll have multiple, in this case here, you'll have multiple codons code for a specific amino acid. And here we've got a heap, and here we have a heap. But they all have about 3 or 4 that code for the same, 3 or 4 different codons that code for the same amino acid. So how do we interpret this code? So we can see in our body that this is the whole entire process. So we have transcription occurring here to get our piece of mRNA off our template DNA strand, and it's interpreted in groups of 3 called codons. And so in this case, we have a U, a G, and a G. So how we look at this on our sort of chart here is we first look for our first base. So our first base we can see is a U. So we can find that a U is here. Our second base is a G. So we look in this G. So now we know that we've narrowed it down to somewhere in here. And with the G, we know that it's narrowed down to here. So now we've got UGG again. So that means we know it's this one, the third base, UGG, which is this same one here. Same for phenyl, the PHE. I won't say the full name. You don't need to know the full names. But we've got UUU. So the first base we'll find is U. Second base is U. And third base is U. So we've got UUU right here. So we've got the PHE. So that's sort of how you do it. So let's have a go ourselves. So we will break these up into the codons. So we break them up into the triplets so we can read them a bit easier. So we can see we've got 4 different codons here. The first one, AUG. So we look for A, we look for U as our second base, and then for our third base, we look for G. Oh, I've gone too high. We look for this G here because it's in this room. And so what we find is that this is the start codon. So it tells it to start, but we can see it also tells it to make an MET. So it says MET is what we want to make, and it puts this amino acid here. So that's the amino acid right there on the drawing. Now we move on to the next one. So we've got this amino acid attached, and now this will move along that way, and we'll read this one now. So we've got AUC. So we start again, we look for A as our first base, then we go to our second base, which will be U, and then our third base, which is a C down here. And the reason why we look at this C here and not this C or this C is because it's in the same column as this first base. So we've got AUC, and we see that that codes for ILE. So ILE, we'll draw that as a triangle. ILE adds together. Awesome. So now the next one is UCG. I think you guys might be starting to get the hang of it. So you can do it the same way as by looking for the U, and then the C, and then the G. Or you can just scan this entire thing and find that UCG is right here. So what we have is that SER. So we'll draw that as a square, SER. And so these are the amino acids joining together, which the ribosome is helping do in our body, but right now I'm the ribosome working it out. So the next one is UAA. So we find our U again. We find that we're in A, so that we know we're in this block somewhere. And then our A again is here. Oh, look, it's told us to stop. So this has said stop. We're not going to add any amino acid here. And now we are left with our final amino, our peptide chain here, our protein. That is our protein right there. So in summary, guys, the mRNA leaves the nucleus, and then the mRNA binds to a ribosome, which we can see here. It's left the nucleus, and it's about to bind to a ribosome. So now it's bound to a ribosome. And what the ribosome is going to do now is going to read it in groups of 3 called codons, which will code for a specific amino acid. And it begins linking these amino acids together to form a protein. So we can see this happen as it moves along. The protein chain gets larger and larger, and then eventually it'll reach a stop codon, which tells it to stop making the protein. And just so you guys know, you don't have to remember the codons because we've got those charts like on the last slide that we can refer to always. So as long as we can just sort of identify this process, this is what I want you to get out of today's lesson. This is everything I want you to remember is right here. And so this entire process is called translation, not transcription. Transcription is from DNA to RNA, whereas RNA to a protein is translation. So great work, guys. Have a go at the questions. I look forward to seeing your responses.